All right, and then I got cut off, but lastly, I will be determining the effectiveness of the kinase inhibitors on tau phosphorylation. So I'm going to perform the above experiments, and then I will add in the inhibitors during the transfection step. I'll be using flow cytometry across a wide variety of concentrations of the inhibitors to make sure I'm not killing all the cells. This will help me determine the IC50, which is when half of the cells survive but half of them die. This is considered the kind of bridge point for safe treatment. Then I have my aim 2. This is to identify the kinase that is phosphorylating threonine 231 and tyrosine 18, as well as the timing in relation to other sites. So the first thing I'm going to do is generate a transgenic mouse line. So my plan for this is to inject white mouse embryos with a vector that contains the kinase that I'm interested in, the PDGF promoter, and a black coat color tag. And then you can also see the homology arms in part A. I will, after injecting the mice, the offspring, this mouse should become half white and half black. And then I will cross these mice, and you can see this in part B of my figure. And simply due to Mendelian genetics, I should get about 25% of their offspring being all black, which indicates that they have the kinase that I'm interested in. I will then perform a time course analysis on the tyrosine 18 threonine 231 phosphorylation. My plan for this is to obtain hippocampus samples at 1 month intervals over a period of 18 months. At each one of these month intervals, I'm going to take 3 to 5 mice, then dissect and lyse out the hippocampus, purify tau as I described in AIM 1, and then run a gel with phosphospecific and pan antibody. For these two sites, the later sites that I mentioned, and alanine 16 as my control site because this will not be phosphorylated. This will help me understand the relative amount of phosphorylation at any given time in the mouse. And then from here, this will help me understand the treatment and how we can administer these inhibitors over a certain time frame. This experiment should be able to help me also understand what kinase is being most active, and then I can administer that inhibitor to these mice. That would be a later step. I'm not as interested in doing that right now, but that is something I would be interested in doing from this aim. My third aim, this one I'm actually super interested in. I read a whole bunch of papers and I couldn't find anything on it, but I'm interested in looking at if the hyperphosphorylation of tyrosine 18 and threonine 231 are both necessary for the phosphorylation of the later sites that I mentioned. So the serine 202, threonine 205, and then serine 199, 422, and 396. For this experiment, my plan is to run kind of two parallel experiments. So the first one, I will be using the previously generated mouse line that I had in AIM 2 to determine the phosphorylation relationship. Here, the mice will be given the inhibitor of the kinase that they're overexpressing, and then I'll do the same time course analysis as I previously described on the hippocampus, lyse out the tau, run the gel. Then the second experiment I'll be running is I will actually generate a new mutant transgenic mouse line. Here they will still have the homology arms, a coat color tag, this time it'll be gray, and then the PDGF promoter, but instead of the kinase, these guys are actually going to be expressing the mutant tau. The mutant tau is going to have alanine transition at the tyrosine 18 and threonine 231. This should prevent the phosphorylation of these sites entirely because alanine lacks the OH group, it just has a CH3. The mice that I get once I inject the embryos will probably be heterozygous, so they'll be both gray and black, and then crossing with Mendelian genetics should give me 25% of my offspring that are all gray, which indicates that they have both the mutant tau and they will also have the expression of the kinase from the original mouse line. So the reason why I'm doing this is because I want to run these parallel to each other because this first experiment is something that we could physiologically do to the human body. We can give people drugs, we can give them an inhibitor of a kinase. But the second step I'm kind of using as a proof of concept. So these, I know for a fact that they will not phosphorylate at these two sites. So I should be able to compare the phosphorylation of these mice to the previous mice. And then my analysis of the results. The result that I would be really interested in seeing would be if the later sites were not phosphorylated. This would indicate that phosphorylation of these initial 2 sites were necessary for the phosphorylation of the later sites, which then induces the progression of Alzheimer's disease. That would be a really interesting result. Is that the result? I don't know, but it would be pretty cool. So the conclusions that I plan on taking from my experiments, first, I determined the kinases that are especially active in tau phosphorylation. This has potential to be a target for treatment, and it's also relevant to anyone that's working with tau or proteins that are phosphorylated in the brain. Treatment options for not just Alzheimer's disease, but Parkinson's and others as well. I would also be interested in determining the timing of the early phosphorylation. If I can better understand when these phosphorylation events occur, I can treat patients more effectively. Because if I can extrapolate back from the mice, their age that they were phosphorylated at a certain point in time, back to humans, I can know whether or not I can administer a certain drug at a certain point in time, which is really important in doing translational research. Lastly, out of just curiosity, I'd like to understand the relationship between the phosphorylation sites. So as I just said, if the phosphorylation events at tyrosine 18 and threonine 231 are needed for the phosphorylation of the later sites that I mentioned, serine, 202, etc., if we could prevent the phosphorylation at these 2 sites, this should either slow or halt the progression of Alzheimer's disease, simply because the tau protein can't continue to be hyperphosphorylated, and therefore it can't misfold and interact in an improper way. Future directions that I plan on taking my experiment, I'd like to study the structure of mutant tau, simply because unless I'm really bad at Googling, I didn't really see too much information on what happens as the sites become phosphorylated and how this impacts the structure of the protein. And I'd also like to look at the pH of the environment around the protein as the proteins are phosphorylated. These 2 pieces of information together could help me determine whether or not it's the change in the environment or if it's the change in the conformation that causes the later phosphorylation sites to occur. For example, if the conformation changes, the kinase might be able to fit into the site easier and then phosphorylate those amino acids. Or if the pH changes in just the right way, the kinase may be able to function more effectively. This is just hypothetical, I'm not really sure, but those are 2 areas that I'd be interested in looking. Okay, frequently asked questions. You guys had some really awesome questions in the grant comments. Were very thought-provoking. I tried to answer some of them in my presentation. There are a few questions on the NT2 cells and some transfection and experimental approach questions, so I hope that I addressed those. If I didn't, let me know. You can email me or something. But these were a few of them that I thought were super interesting. So, couldn't inhibitors for tau be introduced to prevent the aggregation of tau proteins, or would this inhibit normal tau function as well? This was actually my initial thought process. I thought about just knocking out tau entirely and seeing what happened, but tau is so critical in normal microtubule function that inhibiting it would actually have worse effects. Tau keeps the microtubules functioning properly, and microtubules functioning properly is so important for cell survival and synapses and transport within the cell. It's just too risky to knock down tau entirely as a treatment option. Then have you thought about examining the cerebrospinal fluid of the mice to observe the tau levels, since they tend to be elevated in Alzheimer's disease? Yes. This was another option that I considered. I didn't think I should have 6 aims, but I did choose to stick with the brain because I really wanted to understand the structural impact, especially in the hippocampus, because those kinases, like the MARC kinase, functions almost, not exclusively, but really powerfully within the hippocampus. I wanted to see what the effects were there. Also, I like the Braak staging analogy, how it had the increase in tau phosphorylation and in the different structures throughout the brain. Cerebrospinal fluid is something that I think would be a really good next step for any and all of these experiments. Okay, this was a great question. Is the hyperphosphorylation of tau referring to the increased phosphorylation sites or the increased amount of time spent phosphorylated? It's both, kind of. So the sites that I have been mentioning, so the serine sites and then the threonine, and tyrosine sites that are the earlier sites, these are not normally phosphorylated in tau, so yes, there is increased phosphorylation. But as you can kind of see in the Braak staging, the longer the sites are phosphorylated, the worse the prognosis is for the patient. So if you're in stages 1 and 2, you only have a little bit of phosphorylation in one area, but as you progress through stages 5 and 6, not only do you have more places that are phosphorylated, but those original sites that you were already phosphorylated have been phosphorylated for a longer amount of time. So it's bad. I hope that explained it a little bit, but it is both really good question. And lastly, thought-provoking, what if I didn't see the results I expected in specific AIM-2? Would I continue to use the same mice for AIM-3? So here I would just use those new transgenic mice that I make in AIM-3 as a proof of concept. I really want to understand whether or not the later sites need that earlier phosphorylation. The reason why I used the mice from AIM-2 was because I wanted to be able to do something that was applicable to humans, but having just that proof of concept of using the mutant mouse line that has those alanines is also fine for me. But yeah, that was a really good question, and yeah, proof of concept, just understanding what would happen. And that is all I have for you guys. Thank you for listening, and I hope you learned something. And if I didn't answer a question, just let me know. I hope you all stay safe and healthy. Bye.